everyone, welcome back to this part of video presentation. Here we will discuss the transcription mechanism in eukaryotic system. In the previous discussions, we have already seen that there are many proteins, different sequences and certain regulatory sequences and regulatory proteins which are associated with the transcription process. And even before that, we discussed about the bacterial mechanism and also the bacterial machineries involved in transcription. So we also have a comparison between the prokaryote and eukaryote in terms of the proteins and sequences associated, and also in the mechanism itself. And when we compare the mechanisms in prokaryotes and eukaryotes, the main difference comes in the initiation process itself. And also there you can see a kind of difference in the termination event also. So all those things we will discuss in detail in this part. So let's move to the presentation side. So let's begin the presentation with an introduction to the RNA polymerase enzyme. As already we know that there are three types of RNA polymerases. Hope you know all this. We have RNA polymerase 1 for the synthesis of rRNA molecule, and they are usually located within the nucleolase. Right, I'm not going into the what is a nucleolysis. The second type is RNA polymerase 2 for the mRNA synthesis, and commonly seen in the nucleoplasm where the transcription is located. And the third type is RNA polymerase 3, which is also in the nucleoplasm for the tRNA and some small rRNA synthesis. And these are the sequences associated with a gene which is going to be transcribed. So these include the co-promoters, the accessory promoter sequences like the regulatory sequences, maybe it's an enhancer sequence or a proximal promoter element, and there are sequences which positively regulate the transcription, and also there are negatively or down regulating sequences. So along with the events which are regulated by all these sequences, the transcription will continue. And here this is about the enzyme and the transcription factors. We have the different transcription factors as already we have discussed in the previous video. This is the order by which they are involved, like all these factors like transcription factor D, A, B, then F, E and H, all are associated with the transcription machinery. The structure what we are seeing is nothing but what we call the closed complex or the pre-initiation complex. So these are the three important factors which need to be in mind when we are going to the mechanism of transcription in eukaryotic system. So again, the quantity of the factors are given there and also their corresponding functions. So as already we have seen it previously, I am not moving into that in detail. So as for the other mechanisms and as in prokaryotes, the transcription in eukaryotes also we have divided into three phases. One, that means initiation, elongation and termination. This is for our understanding of the mechanism for easy discussions. Right, so initiation in turn we can see certain mechanisms. So again we have divided them into different stages. So we'll start with the first event, that is the assembly of RNA polymerase and the transcription factors at the promoter site. We know definitely that will be the first process. That means we need to identify the site where the transcription has to commence. So that's actually communicated through the transcription factors. So the transcription factors will play a crucial role in recognizing the site of transcription and that recognizing part of the chromosome is what we call the promoter sequences. So here the first transcription factor which is going to interact or which is identifying the promoter sequence is what we call the transcription factor 2D. And remember, we are discussing all these mechanism in terms of the RNA polymerase 2, and hence we are using the transcription factors as the transcription factor 2 which is associated with the RNA polymerase 2, right? So the TF2D will go and bind with specific sequences in the chromosome, what we call the core promoter region, or even specifically you can say the Tata box, which are located at around -25 in the upstream site. So in turn the TF2D consists of, as you know, the Tata binding protein and the Tata binding protein association factors, TAFs. So with the help of the sequence in the TF2D, what we call the TBP, Tata binding protein, this protein complex will go and bind to the Tata box. That is the first element which is binding over the promoter region. And also in most of the situations, the TAFs also will facilitate interaction of the proteins into the promoter region. And even in a sequence or in a promoter without a Tata box, the TAFs will support the interaction. And now the order of event, the eventual mechanisms are like the next transcription factor to come into place is TF2B, which usually recognize a different site, what we call the BRE, TF2B recognition element, which is still upstream to the Tata box, usually at around between 30 and 40. And this is simultaneously, there is another factor called TF2F, which is also recruited to this complex. Then now it will become a complex of TF2D or the TBP, TF2B and TF2A complex. And the TF2B and TF2A support the system to interact or to create a stable interaction with the promoter sequences. And only after a stable complex has been formed, only the next factor, the most important factor, what we call the RNA polymerase enzyme, will attach to the site. So they will stabilize the structure. So the factors like B and A are important for a successful transcription initiation to commence. Now the next element, that is the transcription factor 2F, will come along with the RNA polymerase 2, the enzyme involved in the transcription process. So the RNA polymerase enzyme can identify the promoter sequences, but with the help of the transcription factor 2F. So they come as a complex, right? So now we have the next factors are, once all these factors like Tata binding protein, TF2B, then Pol F complex is formed, we are moving into the last phase of the complex attachment, that is the E will be coming to the complex next, and E will facilitate the binding of next factor, what the TF2H, which is a significant molecule for initiation process, right? So this is the events which are happening during the complex formation of initiation, or what we call the pre-initiation complex, or it's otherwise also called the closed complex, right? So this will essentially will cover the +1 site, that means the site where the transcription begins. So this is the events. Initially the TF2D comes, then A also will follow that, then there is B, then TF2F and Pol enzyme complex, then the E and H. So they form the closed complex in the system. So the first one is the assembly of the transcription factors and the polymerase enzyme to the promoter site. And the second event in initiation is the promoter melting. right? So we are moving to the second mechanism. Here, promoter melting represents the unwinding of the DNA duplex at the promoter region. So we know that that's an essential part that's required for availing the DNA template for the RNA polymerase enzyme. As we know that only one template on one strand will be acting as the template, right? So actually for this process, the helicase activity of the TF2H will support the system, and this causes melting of pretty length of a DNA duplex. Usually around 15 to 18 base pairs will be unwound, and which will be spanning the +1 site. So the +1 site will be open and ready for transcription. That means ready for the addition of nucleotides complementary to the sequences in the DNA template, right? And the energy required for this process will be provided by the ATPase activity of the TF2H itself, right? So you can see an open region within the transcription promoter region, and now the system has moved from the closed promoter or the closed complex to an open complex. Now it's ready for elongation process, right? But still we have some other processes also we have to see. And when the first nucleotide is added to the site, so as we know that the RNA polymerase doesn't require a primer, so the first nucleotide could be added by the enzyme itself. So the first nucleotide will be added to the +1 site, right? So into that the remaining nucleotides will be added as a usual process. There will be nucleotide addition by the RNA polymerase enzyme, and that is nothing but the catalyzation of the 3' OH and the 5' incoming 5' phosphate will be attached through the phosphodiester bond formation. So this will continue for around around 9 to 12 nucleotides. So once it reaches around 10 to 12 nucleotides, the region of the RNA polymerase will over. That means the bound region will be over. Now what they do is they'll try to move, the RNA polymerase will try to move in the forward direction so that the polymerase can synthesize the next part of the chromosome. But this will not be allowed by the system in the beginning, and for that certain conditions has to be satisfied. So which are these conditions? The first thing is there are certain hindrances, there are certain blocks in the system itself. And which are they? So the first thing is that the TF2B usually occupies an active part of the active site of the enzyme, and which will block the elongation process, and that has to be displaced. And the second thing is that the exit channel of the enzyme, the exit channel, that means through which the synthesized RNA molecule will come out of the complex, is also being partially occupied by the transcription factors. So all these blocks has to be removed, and before the removal of these processes, the synthesized RNA molecule cannot be elongated. So certain short stretches of RNA molecule will be synthesized, and that will be eliminated from the system. And this event is what we call the abortive initiation. And in a eukaryotic system, we can see a number of cycles as abortive initiation, and only after a number of immature or short stretches of RNA has been synthesized, the system will move into the elongation process. Right, and the fourth phase is the promoter escape. That means that is the successful promoter clearance by the RNA polymerase enzyme. Right. Okay, so RNA polymerase enzyme moves along the has to move along the DNA template. So once it is forming an open complex, then it will appear like a transcription bubble. Already we have noted previously. So this transcription, within this transcription bubble, we know that there will be the DNA template, the newly synthesizing RNA strand, the enzyme system, and also the transcription factors, even if it is in the initiation phase, right? So the TF2B is an important factor which hold the complex within the transcription bubble in the initiation stage. That means this TF2B can bind onto the DNA and RNA for stabilizing the structure. And if you want to move away from the promoter region, you have to dissociate, or you have to cut the bond between the TF2B and the DNA RNA hybrid, or between the TF2B and the DNA itself, or the promoter complex itself. So this mechanism is what we call the promoter escape, right? So the promoter escape is a movement of the transcription bubble leaving the promoter site and the most of the transcription factors. So let's see how this happens. So this is the event. You can see in the image all the transcription factors are left in the promoter region itself, and you can see that these will be utilized for a next round of transcription within that region itself. When you are there, that sequence is loaded with an RNA polymerase complex again, right? So the promoter clearance require a most important reaction, what we call the phosphorylation of the carboxy terminal domain of the largest subunit of the RNA polymerase 2 enzyme. That's what we already noted. It is RPB1. And this RPB1, again, we have seen before, there is a heptapeptide sequence which consists of certain serine residues. So these serine residues are usually undergoing heavy phosphorylation by the involvement of TF2H kinase activity, and also certain other kinases are also associated. So you have to note that in the beginning, or in the initiation event, the CTD of the RPB1 will be in the unphosphorylated stage, right? So only when it is in the unphosphorylated stage, the initiation is possible. And once the initiation is done, now it is going to phosphorylate, right? And this is done by the TF2H. And once the phosphorylation of the CTD of the RPB1 is done, this causes a dissociation of the enzyme from the transcription factors, especially from the TF2B and other transcription factors. And this will cause the polymerase to move along the RNA polymerase. Move along the DNA template. So these are the events initially we know. So this is a consolidated structure. So you have the start site. Now the TBP comes, all the other factors, B comes, A comes, then the polymerase enzyme forms the complex. Then this is the initiation complex, and after that we know the H will utilize the helicase activity to form the open complex, and then once open complex is done, there is promoter escape with the help of again TF2H kinase activity, right? So these are the events in the initiation. Hope you understood about the initiation event. So we have seen the importance of additional factors in the transcription process. Like for the opening of the condensed chromatin, we need these activators, mediators, etc. So they will promote the mechanism to occur as in a fast rate. So I am not moving into that part of the discussion. So the second phase of transcription is elongation process. So in elongation, the RNA polymerase II enzyme will move through the region of the gene, what we call the template DNA, right? So after the successful initiation, the RNA polymerase will be moving along the template, and there will be addition of nucleotides complementary to the template, and this occurs as a 4-step reaction. First, the nucleotide complementary to the template sequence will come into the entry site as an inverted molecule, and after that there is the position that will be corrected in such a way that will be oriented into the correct orientation, and also it will be undergoing a cross checking whether this is the correct base which is required to be added over there, and also it will check whether it is the original native one, that means whether it is the DNTP or ribonucleotide or deoxyribonucleotide, right? So after that checking is over, there will be the phosphodiester bond formation, and finally that cycle will repeat. So these are the events which is happening during the elongation process. And in the upstream end of the hybrid, that means in the bubble, the RNA polymerase II separates a nascent RNA from the DNA. That means the newly synthesized RNA will be dissociated from the hybrid at the upstream end of the hybrid structure. You can see this. So there is once the CTD has been phosphorylated, you can see they also use the energy for the movement with the phosphorylation of with the energy from the ATP molecule, and there will be elongation, and newly synthesized RNA will be coming out of the complex, right? So the involvement of mediators and activators already we have seen, but still we have to see another thing, that all these mediators and activators, what they do is they decondense the chromatin up to the nucleosome level. So but that's not enough, right? We need a bare DNA, that means we need a free double-stranded DNA molecule as a template. So how these nucleosomes, when present, the transcription is going to happen, right? That's a question. If there are nucleosomes present in the system, in the DNA molecule, then how the RNA polymerase can move along, right? So that's an important question. So for that we have certain mechanisms. So already we know that a nucleosome will be covering somewhere around 200 base pair distance. So that region, before the polymerase is coming into that region, has to be cleared. That means that region has to be evacuated for the polymerase to go with the successful transcription process. So how this happens? There are two mechanisms proposed. One is the nucleosome mobilization, in which the nucleosome itself will move from an untranscribed region to a transcribed region. There will be a displacement, what we call the nucleosome mobilization. This image will show that mechanism. Already you can see that that blue as the nucleosomes. So the nucleosomes will be moving into a transcribed region from an untranscribed region. The second mechanism is that a removal of the octamer core, that means H2A, H2B dimer removal. That means the nucleosomes will be disrupted during the movement of the enzyme, and that will be followed by the followed by the transcription process. And for that, certain factors are helping in the process, and after that these dissociated molecules will come back to reform the chromatin structure, right? So finally we are moving into the last phase called the transcription termination process. So hope you understood about the initiation. There is initiation and followed by elongation. Elongation will continue to a particular point where we are going to stop the transcription process, right? But unlike in prokaryotic system, in eukaryotes we do not have a specific sequence or any specific signals are present in eukaryote, right? So there is no such sequences to terminate in eukaryotes, right? But how then it occurs? So the RNA polymerase will continue to transcribe along the template, and the interesting fact is that RNA polymerase will be synthesizing even covering the end site, which covers thousands of base pairs which are not required, right? So even covering the end site, the transcription will be continuing, and after that the transcribing or the terminating systems will be activated, and that will cut the RNA in the downstream of the newly synthesized, in the upstream of the newly synthesized RNA molecule, right? So and after that, so once you can imagine, like, there is an RNA which is being synthesized, and at the downstream end or at the 3' end, then RNA will be synthesizing in the complex, and that will be cut at particular point within that, and that will be released out. So this will be continuing the synthesis, and this synthesis will be blocked with the binding of an exonuclease to this end, and that will cleave this complex, and this will be subjected for further processing, right? So we'll see, this is the site. So even though there is no specific sites for termination, we can see that there is a specific site for polyadenylation in the pre-mRNA molecule. You know what about polyadenylation? We will discuss that in RNA processing presentations. So polyadenylation is a important event which is present in all mRNA molecule, right? So for the polyadenylation, there is a sequence which is specific for polyadenylation, and these are denoted as the AAUAAA. You can see that. So the cleavage site, which determines the end of the emerging pre-mRNA, occurs usually between the upstream AAUAAA sequence and the downstream GU-rich sequence, and these two sequences will be separated some around 40 to 60 nucleotides in the RNA molecule. You can see that. So this is the AAUAAA sequence at particular site, and in the downstream to that you can see a GU-rich region, and the cut site usually comes in between. So that will be the cleavage site. So here the cleavage usually occurs. So you have to remember that this is not the termination sequence sites at all, but these are the polyadenylation sites, or the polyadenylation marking site, right? So in the next phase you can see, these part will be transcribed. Once these transcription is completed, certain external proteins called CPSF will bind to the AAUAAA sequence. CPSF is nothing but cleavage and polyadenylation specific factor. So this CPSF will bind to the AAUAAA sequence, and another protein called CstF, it is cleavage stimulating factor, binds to the GU-rich site, and then these CPSF, and also there are certain cleavage factors. It's not like CF1 and CF2, they are actually cleaving the RNA molecule. So the cleavage proteins cleaves the nascent pre-mRNA at a site, usually at a site 10 to 30 nucleotides downstream from the AAUAAA site, right? So you have to remember that the cleavage site usually occurs at around 30 nucleotides downstream of the AAUAAA sequence, right? And once it is done, the additional proteins like the poly-A polymerase will also come to this complex for the polyadenylation process, right? Then the remainder of the transcript. So you can see that once it is cut, this segment of the DNA will remain in the template, right? And even at that time also, the transcription is going on in the downstream of that RNA molecule, right? So the remainder of the transcript is digested by an exonuclease enzyme, right? So this exonuclease will cleave the remaining part of the RNA, and it will move along the cleaved, or which it will move in the 5 prime 3 prime direction along the RNA molecule, and will dissociate the DNA RNA hybrid, and that causes a termination process in eukaryotes. You can see that in brief, right? So there is a sequence which is marked for polyadenylation, and there is binding of the specific proteins CPSF and CstF into these sequence. Then this is followed by binding of cleavage factors or the cleavage proteins, and also there are many other proteins also. And then there is cleavage, and once the cleavage is done, there is binding of poly-A proteins and other associated proteins required for the polyadenylation process, and there will be polyadenylation going on. You can see there are addition of adenine residues. There usually around 80 to 250 adenine residues will be added at the end, and so that's all for the termination. So I hope you understood about the mechanism in eukaryotes. We have initiation, elongation and termination. In the initiation stage, all the transcription factors, the enzymes will occupy the promoter site. There is promoter, there is closed complex formation, then followed by open complex with the help of TFIIH. Then there is an abortive initiation, then followed by promoter escape, and promoter escape is usually is facilitated by the TFIIH kinase activity, and there will be a phosphorylation of the CTD domain of the RPB1, and this is followed by elongation. So there is elongation in which the nucleotides will be added complementary to the template DNA, and this will continue and until there is a cleavage occurs, right? So the cleavage occurs, that's what we demarcated as the termination. The cleavage occurs at a site between a specific sequence called the AAUAAA and a GU-rich sequence, somewhat 60 base pairs downstream to the AAUAAA sequence. So to that region, certain proteins will occupy, and there will be cleavage in between these two, usually around 30 base pairs downstream to the AAUAAA sequence, and the remaining sequences will be cleaved by an exonuclease activity, right? And the synthesized pre-mRNA will be subjected for the next round of processes, what we call the post-transcriptional processing. So that we will discuss in the next videos, and I hope you understood. Thank you for watching.